Hello, everyone. Welcome to a webinar, part of the LabRoots Coronavirus Virtual Event Series. My name is Elizabeth Schneider. I'm a Senior Global Marketing Manager here at NanoString. Today, we have the pleasure for the first presentation of this coronavirus virtual event to have two speakers here today to talk about tackling COVID-19 by looking at temporal and spatial expression of infected tissue and infectious samples from patients and convalescent individuals. Up first today, we have Dr. Eugenia Ong. She is a Senior Research Fellow at the Viral Research and Experimental Medicine Center at SingHealth Duke-NUS, the acronym ViREMiCS, in the Duke-NUS Medical School. And she works on the development of virological, immunological, and genomic assays to determine the molecular correlation for preclinical development and efficacy monitoring of therapeutics and vaccines. And she will be speaking today on the temporal dynamics of the host molecular response underlying severe COVID-19 progression and disease resolution. We will be collecting questions throughout the presentation, and we will pause before we move on to our next speaker to take the questions for Dr. Ong. So please go ahead and submit your questions throughout the presentation, and then we will hold them until the end for the Q&A discussion. And with that, thank you very much, Dr. Ong, for being here. We're really looking forward to hearing your presentation.
Yeah, it's my pleasure, Elizabeth. So a large thank you to NanoString and LabRoots for providing this platform and opportunity for us to share our work today with you. The work that I'm going to be describing to you today is on the temporal dynamics of the host molecular responses that underlie severe COVID-19 progression and disease resolution. And this work was recently published in eBiomedicine in March 2021. And so I think COVID-19 is no stranger to anyone who is on this webinar today. To date, there have been more than 135 million COVID-19 cases worldwide, with deaths arising from COVID-19 creeping towards 3 million cases worldwide. And so since the COVID-19 pandemic started, I think multiple groups around the world have been trying to look at the underlying immune factors for COVID-19 severity. And so here, what we've done is we've summarized the events that happened during both the acute phase of illness as well as recovery phase to give you just a flavor of the types of immune factors that could underlie COVID-19 severity. And so in the acute phase of disease, what multiple groups have found is that the host immune system fails to launch a robust type 1, type 3 interferon response. We also see increased numbers of inflammatory monocytes, neutrophils. There is some degree of lymphopenia as well as exhausted T-cell responses, as well as increased levels of pro-inflammatory cytokines such as IL-6 and interferon gamma that could be predictive of disease severity. In contrast, during the recovery phase, we see recovery of T cells, including memory CD8 T cells, as well as the recovery of dendritic cell subsets. There is also reduced numbers of neutrophils, as well as increased basophils and eosinophils, as well as a reduced level of IL-6 interferon gamma, as well as CXCL10 during the recovery phase. However, seeing the causative from the associative factors of respiratory dysfunction remains an urgent unmet need. The host responses that cause respiratory function, we think, would track most closely with disease progression and resolution, and therefore permit differentiation from other factors that could be just statistically associated with, but not causative, of severe COVID-19. And therefore, the statistical association with COVID-19 could also be heavily influenced by sampling points in the course of acute illness. And so in our research, what we've done is to focus on the host response to SARS-CoV-2 infection around the period of the nadir of respiratory function. And we sampled daily from COVID patients whenever possible. And so our hypothesis is that the principal drivers of COVID-19 severity should track tightly with respiratory function and should also resolve in sync with respiratory improvement. And so you'll see that on the table on the left-hand side is a table showing clinical features of patients that were included in our study, consisting of 10 patients, 8 male and 2 female patients. Patients range from disease severity from mild to moderately severe to severe. And on the right-hand side, you'll see an overview of the sampling points that were taken from these patients over the course of disease. And this ranges from around -4 days to 13 days post-nadir, with day 0 being the day of nadir. For most of these time points, you'll see that we would have done sampling that we then eventually sent for both microarray as well as nanostring profiling. And so we use oxygen requirement to stratify patients into mild, moderately severe, or severe COVID-19. And of the 10 patients, 4 were classified as mild, as they did not require supplemental oxygen. And for the remaining 6 patients, for which you can see their clinical overview on the left-hand side of the screen, all patients here required supplemental oxygen, and 5 patients required more than 2 liters per minute of supplemental oxygen, requiring either mechanical ventilation or the usage of high-flow oxygen devices, while the remaining patient, who was moderately severe, received not more than 2 liters per minute of supplemental oxygen. The severe COVID-19 patients also displayed leukopenia, as shown on the chart on the right-hand side, despite normal white blood cell counts, and this is also consistent with findings from other groups. So we sampled blood daily from patients where possible, and this was followed by RNA extraction from whole blood before the RNA was then sent for microarray profiling. And so on the left-hand side of the screen, you'll see a waterfall plot that shows differentially expressed genes that were detected over the course of disease in patients. And these differentially expressed genes are expressed with a fold change of more than 1.5. With an adjusted p-value of less than 0.05. When we conducted functional annotation of downregulated genes, it revealed that the immune pathways were the most significantly reduced with time, as shown on the plot on the right-hand side of the screen. And this included pathways such as cytokine and interferon-dependent immune responses, inflammatory signaling, as well as neutrophil-mediated immune pathway to be most significantly reduced with time. In contrast, we did not find significant pathways among the upregulated genes. We next validated changes in the expression of innate and adaptive immune genes using nanostring profiling. And so conducting a principal component analysis confirmed that COVID-19 patients segregated distinctly from healthy controls, which consisted of baseline samples that were obtained from participants of a measles, mumps, and rubella revaccination study. So we can see that the top contributors in PC1 consisted of genes that were involved in toll-like receptor signaling as well as inflammatory responses, and this was important for distinguishing the mild from the severe patients. In addition, the top contributors in PC3, which differentiated healthy controls from patients with COVID-19, included genes that were driving the cytokine as well as the interferon responses. From nanostring profiling, what we found was that the top downregulated genes included pathways such as toll-like receptor as well as the type 1 interferon signaling pathways, which was consistent with what we had found with microarray. In addition, the immune pathways that were upregulated during the recovery phase included pathways that could be related to T-cell proliferation and activation. Interestingly, as you see on this slide, when we plotted out these gene expression profiles temporally, the expression profiles for these pathways suggest that suppression of the type 1 interferon and toll-like receptor signaling pathways actually precedes restoration of the T-cell proliferation and activation pathways. And the heat maps on the screen right here reflect this very clearly. So to determine the genes that track with disease progression and resolution, we next use extraction and analysis of differential gene expression, or EDGE analysis, to obtain greater insight on the temporally regulated transcripts over the course of illness in severe patients. So from EDGE analysis, we identified 4,831 temporally regulated genes, which following that were put through self-organizing map clustering. And through this process, you'll see that these genes, there's actually distinct transcriptional dynamics that are shown in severe patients, and they happen to segregate into two distinct modules. So genes in module A are shown to be decreased post-nadir, whereas genes in module B are increasing post-nadir. And so we think that combining EDGE as well as self-organizing maps clustering, or SOM clustering, allowed us to leverage both the clinical as well as temporal covariates for increased statistical power to identify genes that track with disease progression as well as recovery. So if we were to perform a pathway analysis of genes that were shown to be decreasing post-nadir, what we identified was that genes are implicated in cellular response to type 1 interferon, the regulation of immune effector functions, as well as neutrophil-mediated immunity. So of note, I like to focus on the pathway for neutrophil-mediated immunity, which is the most significantly enriched pathway found in clusters 3 and 4. And as you can see from the bar plots on the right-hand side of the screen, at the bottom right-hand side of the screen, the gene expression profile for genes that are driving neutrophil-mediated immunity seem to track very well with both disease progression as well as disease recovery. So of note, neutrophil activation as well as the presence of neutrophil extracellular trap markers have been observed in the lung of COVID-19 patients in postmortem examinations. And besides COVID-19, neutrophils have also been found to be enriched in the lungs of patients with acute respiratory distress syndrome caused by other infections such as influenza. And we think that because of this context, it provides a greater clinical relevance for pathways that have been identified in our analysis. So significant pathway enrichment was derived from genes in cluster 6. And so these are the genes. So next, what we focused on were genes that were shown to be increasing post-nadir. And we found significant pathway enrichment only derived from genes in cluster 6. And so in these genes, what we saw was an increased protein phosphorylation signature in COVID-19 patients that were recovering from severe disease. And so of the 36 genes that were significantly regulated in the protein phosphorylation pathway, 20 were found to be serine threonine kinases, as shown on the heat map on the right-hand side of the screen. Interestingly, when these genes were cross-referenced to RegFos, so RegFos is a database that allows exploration of the human protein phosphorylation regulatory network. We found that these genes were involved in cell growth, cell survival, as well as T-cell development, suggesting that kinases could play a protective role in disease resolution. And indeed, we think that perhaps protein phosphorylation could be key to restoring T-cell numbers during the recovery of illness, as we had shown earlier in the heat map from before. Now, since genes in cluster 3 and 4, or those that are responsible for neutrophil activation as well as inflammatory responses, are strongly associated with disease progression and resolution, what we did next was to cross-reference these transcripts to the drug-gene interaction database, or DGIDB. And the candidate drugs that you see on the left-hand side are a result of that analysis that was done. So the drugs that target genes in cluster 3 and 4, or genes that are decreasing post-nadir, are shown in blue. And the drugs that target genes in cluster 3 and 4, but not genes in cluster 6, are highlighted in pink. And this identifies differentially drugs that could target genes that are downregulated post-nadir, but not those that are increasing post-nadir, to prevent any off-target effects. And so the candidate drugs that corroborate with drugs that have shown efficacy outcomes in clinical trials, including dexamethasone and aspirin, are shown highlighted here on the plots on the left. And in contrast, drugs with no efficacy in improving clinical outcomes, such as lopinavir, ritonavir, as well as hydroxychloroquine, were absent from the hits. So in summary, we think that by performing daily transcriptomic profiling of severe and non-severe COVID-19 patients, our findings reveal that neutrophil activation, but not type 1 interferon signaling transcripts, track most closely with disease progression and recovery. And so drugs that target neutrophil activation could be repurposed for the treatment of severe COVID-19. And collectively, our data serves as a resource for understanding the host pathogen dynamics involved in COVID-19 disease progression as well as recovery. So lastly, I'd like to acknowledge our collaborators and colleagues at both the SingHealth Duke-NUS Academic Medical Center, ViREMiCS, the Singapore General Hospital, as well as Duke-NUS Medical School. We thank all doctors and nurses in the isolation facility at the Singapore General Hospital. And this study was sponsored in part by a generous grant from the Hourglass. And I thank you for your attention and your time.
Thank you so much, Dr. Ong. That was a really insightful presentation. So a couple of questions came in throughout the course of the presentation. So the first question for you, there are many reports on sequelae caused by COVID-19 in survivors, including loss of hearing, taste, and even psychological conditions. Do you foresee possible blood-based biomarkers that can be developed in the future to predict and intervene with such, symptoms and effects?
Yeah, so that's definitely an interesting question. And the use of blood-based biomarkers, I think, is definitely possible in the long run. We have already done some initial testing using this approach for assessing safety signals for adverse events. For example, our group had actually done a previous study on the yellow fever vaccine, which is actually one of the most effective vaccines to date. And just over 50% of vaccine recipients, they actually report adverse events after receiving the yellow fever vaccine. And adverse events can include symptoms such as fatigue, headaches, as well as muscle aches. And what we showed was that after yellow fever dosing, the activation of innate immune genes at day 1, but not day 3 after vaccination, was directly correlated with the onset of adverse events. So I think that a biomarker approach is certainly possible, but there are similarly also challenges that we have to challenges or practical considerations that we have to take note of. And I think the first one would be how these survivors are recruited, so the recruitment process. And probably this would involve a multidisciplinary team, as you mentioned, these symptoms actually intersect various organ classes. And ideally, I think these survivors would be followed up at primary care providers that could then possibly refer them to enroll in clinical research studies. And the second point of consideration would be how sampling would be performed. And I think this would be the most challenging part, because there is an uncertainty in terms of the time points that need to be collected. And so it's really important, I think, that you have a systematic study design that can capture time points both before, during, and after such sequelae happens, in order to get a good set and a robust set of data for the type of analysis that needs to take place in order to identify these blood biomarkers.
Thank you. So another question came in. Have you done any Tcell receptor based sequencing to look at what types of Tcell receptors are being displayed in some of these patients and individuals, in particular to kind of understand, whether or not the T cells are able to coalesce around a single receptor repertoire, and also what implications that has on, previous, viruses that people who have severe COVID have been exposed to, and whether or not their T cell receptors, quickly recognize the virus, or if they, the T cell receptors have trouble narrowing down on the right receptor that can help combat the virus.
Yeah, so this is another interesting question, and I think it's extremely relevant in the context of the T-cell related findings that we've made, whereby we see that there is a restoration of the transcriptional signatures for genes driving T-cell activation and proliferation. So my short answer is no, we have not done those studies in our lab, but my understanding is that from the literature now, I think there are multiple reports coming out that show both the importance of T cells in mediating protection, both in the vaccine context as well as in a SARS-CoV-2 infection context. Indeed, there was a paper coming out from a group in Duke-NUS Medical School, and this group is led by Antonio Bertoletti. His lab has actually shown that in convalescent blood that was collected from the previous SARS coronavirus 1 survivors, Singapore actually faced a SARS epidemic in 2003. And so this is nearly about close to 20 years after their initial infections. They still had cross-reactive T cells that could protect against SARS-CoV-2. So I think that collectively, this shows that T cells remain a very important weapon in the arsenal of our host defense against SARS-CoV-2.
Great, thank you. And we have time for one more question. Another question came in through the chat. For any of these patients, is there any chance that you might have some tissue samples to look at to try to understand more on the virus side what tissues the virus initially infiltrated? In other words, trying to understand from more of the virus perspective, what route were the individuals infected, by, whether it was, respiratory or gastrointestinal, etc., and maybe how that might affect the outcomes in terms of the severity.
So in the process of collecting these samples from patients, we were actually only able to collect whole blood samples. So unfortunately, we don't really have tissue samples that can be analyzed. But I think other groups have now also performed different types of using different approaches, such as things such as postmortem examinations. And clearly, I think the lungs are always the tissue types that are most severely damaged, both by overexuberant inflammatory response, as well as infiltration of immune cells. And I think that that really sets the stage for the type of infection that SARS-CoV-2 causes, which is one that causes a lot of tissue and organ damage in the lungs.
Excellent. Well, thank you so much, Dr. Ong. Oh, it looks like there's one more question that came in really quick before we wrap up, and I know that you have limited time with us, so I'll see if we can get this question in really quick. So someone asked, how easy is the NanoString nCounter technology to use with biofluid samples compared to, say, doing PCR or RNA sequencing?
Well, so in our experience, I think using nCounter has been extremely useful because it shortens the amount of time needed by a very large extent. And so instead of performing real-time PCR about maybe 5 to 10 times, we were able to save on the amount of RNA that we had in the lab, and use that amount of RNA for just running the nCounter assay once to get a simultaneous readout for over 500 immune genes. And I think that has been very useful in preserving the amount of the RNA that we have remaining from these collected samples, so that we may likely be able to use them in future studies. Yeah.
Excellent. Thank you so much. And I just would like to point out as well for everyone who's joining us for this presentation that Dr. Ong and her colleagues did use the nCounter Immunology and Inflammation gene expression panels. But in response to the COVID-19 pandemic, last year, NanoString did launch a panel specifically for studying the host response to infectious disease, and it's called the Host Response Panel. And we have a human and a mouse version that you can use in your research. Well, thank you so much, Dr. Ong. It was a pleasure for having you. And we will have the next speaker up. All right, everyone. So we're back for our second presentation as part of the NanoString sponsored webinar for the LabRoots Coronavirus Virtual Event Series. So next up, I would like to introduce Dr. GeoMx Yeong. He is part of the Institute of Molecular and Cell Biology, A*STAR in Singapore. So he's an associate faculty there. He is also affiliated with the Division of Pathology at Singapore General Hospital. And today he's going to be speaking to us about understanding the spatial immune microenvironment proximal to SARS-CoV-2 viral antigen and RNA in COVID-19 decedents and survivors by using spatial transcriptomics. And again, we will have a question and answer session at the end of Dr. Yeong's presentation. So please go ahead and put your questions through the chat and we will address them at the end of the presentation. Also, for those of you who had questions for Dr. Ong's presentation and your questions weren't answered, we will collect questions and follow up with them at a later time to address all of your questions. So thank you again, Dr. Yeong. We're very looking forward to your presentation.
Thank you. Thank you, Elizabeth. So thank you for inviting me. I mean, today I'm actually very happy to actually share with you this particular topic about understanding the spatial immune microenvironment proximal to SARS-CoV-2, viral antigen, and as well as RNA in COVID-19 decedents and survival by using spatial transatomics technology. I have no conflict of interest. So, and we all know that nowadays spatial technologies to actually study tissue is a very important things. I mean, it's actually an evolvement from, you can see, initially we only have H&E and IHC under the bright field to study the tissue, and that is actually still a golden standard for the diagnosis for especially cancer. And for the past 5 to 10 years, I mean, the multiplex IHC, which is actually our field, I mean, it's also very hot topic to actually, and hot technology to actually study cancer immune microenvironment or other diseases that require microscopy setting. So this one is actually at the moment is still very important. However, there are many advanced technologies coming out, and all this something that we want to introduce to you today as well. And most of the people also understand that there are other players like Hyperion from Fudai, for example, MBB, all this we will call it advanced technology. They are more useful for research and discovery, and you can see that we actually put them into like involvement that from clinical, translational, and research that based on their cost, turnaround time, and technical barrier. And just to recap, and just in case you're not very sure what is spatial technology and what is the technology that people mostly use, like multiplex IHC, is basically one tissue section that traditionally you only can do one color staining, studying 1 or 2 biomarker. At the moment, you can do easily 6 or 9 or even 50 that claimed by some of the technologies or even more. So, our groups are specialized in this area. If you are interested, please, use your mobile to actually scan the QR code. In here, you can see some of our data that we deposit into the public domain, and you can play with them live and all these things, including this particular image. This is a very interesting platform that we developed. We will talk about it later on as well. So, So what is spatial transcriptomics? Why we are actually using it? So as you know, generally, if you do a bulk RNA sequencing, it generally looks like this. So you are sequencing everything in the tissue. It's like a mix of cells, a mix of different tissue type kind of things, right? So in the single cells era, you can sort the cells. It can run through all the single cells setting, or even without sorting, you run through all the single cell and then you do a clustering. Later on, you can actually study different cell type in the single cell levels. There is there is a relatively a new technology as well, right? So for the spatial transcriptomics, what do we do? Spatial transcriptomics basically just to actually look at all these single cells and to map them back into the spatial information setting. So you can see that you actually know which cell type actually located at where and which kind of spatial morphology and organ setting. And it is actually represent very well the functional tissue. There's a real tissue. The only difference is that the real tissue is a 3D structures rather than a 2D structures. So usually, I'm sure that most of us in here, when we look at a research question or a clinical trial setting, we usually nowadays have two arms, right? One is the tissue arm, as you can see in the top part of the diagram. You have the patient tissue. You are sent through all sorts of spatial technologies, and you have the tissue. You dissociate them into single cells, run through flow, run through single cell sequencing, or you do the same thing for the blood, PBMC, the kind of thing as well. Ultimately, you are thinking whether this can be combined into a model to predict patient clinical outcome, especially in the immunotherapy era. But there is a gap. What is a gap? The gap is that usually people like us that doing the spatial technologies, right? So our clinician friends or our scientist colleague actually have the bulk RNA sequencing data for the tissue, and then they want you to validate this or even produce like a last figure for their paper, or showing that this particular protein also show on the tissues, either in the conventional IHC levels or actually is a multiplex IHC setting. So somehow, not all the time, sometimes you can't see the correlation and concordance. That is actually quite common, right? And I think the spatial transcriptomics really solve this, because you will solve the heterogeneity of the tissue. You will know that whether these RNA, where they are located, and then later on that why are you not seeing this in the protein levels. The other setting is that nowadays single cells RNA sequencing is so very common, and then usually people run through flow cytometry, high dimensional flow cytometry, or side by side, hand in hand, and then to show to the reviewer. sometimes it's correlated, sometimes concordance, but sometimes not, right? And then when there is a little bit of discordance, the reviewer usually will start to question you. I mean, this is we experience a lot, right? So the single cell data, may be not so, real because they are dissociated. Those during the dissociated process, you actually change the biology or immunology of the cells, blah blah blah, and then later on you won't see this in the floor as well. So again, spatial transatomics actually solve this problem, right? Because you remain intact, and then you see these RNA profile signatures on the tissue, and this supposedly need to be more natural than dissociated one. So what is spatial transatomics? In this setting, we are particularly talk about the digital spatial profiling technology from NanoString. So this technology, usually you will stand at the protein levels for about 3 markers. We call it morphology marker, and you use this general marker to actually visualize and navigate under the microscope. Then you know where do you want to capture the gene profiles and signature from the tissue. And then after that, you get the 20,000 genes from the region of interest that you selected, and then you are sent through either the nCounter or NGS setting to actually get the sequencing data. So this is very powerful, and we believe this is a game changer in the field. So to demonstrate how do you use this new technology to actually complement the current setting for other spatial technology, especially multiplex IHC, the kind of thing, we show you a study that we have for COVID-19 story. We particularly look at this in this study. The interesting part is that the cancer patient with COVID-19. So we call it the survival. Some of these patients, they actually survive from COVID, and then later on we get their tissue. So usually how this happened, because they are cancer patients, and then after the recover from COVID, they went to surgery. So we get a surgical tissue. There's one setting, or maybe they went for some biopsy for certain kind of disease. So we get the biopsy as well. So this is question number 1. Number 2 is that we're also interested to understand, the pulmonary tissue compared to the extrapulmonary tissue. So that is also the other thing that we think is very important to understand the immune microenvironment of the COVID-19 setting. So, and how do we dissect this particular scientific question? As you can see, we actually first use the very conventional setting, IHC or RNA-scope to detect where are this virus, antigen, or RNA on the tissue. And then we look at that particular region of tissue to use spatial transatomics to get genes profiles and signature. And the number 3 is to use multiplex IHC to actually prove it. This is also happening in the protein levels. And I'll share 2 stories with you. First of all, first story is the in situ immunity of survival. Focus on the survival based on spatial transatomics. The second part of the story is the paper that we recently got accepted in GUT that is comparing, some patients that they have co-infections of COVID-19 and HIV compared to the COVID-19 alone. This is a very interesting thing. So, back to the story, digital spatial profiling is basically, you can see, for this particular section, this is a colorectal cancer patient that survived from COVID-19. And then you can see we stain CD3 and CD68 just to look at the immune microenvironment, so we know where to actually select the region of interest. And then these are some of the representative images that we actually select the region of interest to get the 20,000 genes. And after we get the genes, profiles of each of these region of interest, we do a deconvolute method like cyber-sort. So every single region of interest, we actually got their cells abundancy proportion of each of this immune cell type. Such as in the cyber-sort we can differentiate into many different subclasses of the immune cells. From myeloid, dendritic cell, macrophages, and also T cell, B cells, and each of these smaller subset as well. And the other beauty of it is that we also detect in situ the COVID virus RNA, so including the spike and also the RF1. Okay, so to cut the long story short, we found that the SARS-CoV-2 genes are very correlated to some of these immune cells. I mean, in here is like plasma cells and Tregs. That is really interesting to us, and why is it, and then whether we can see in other settings. So first of all, we know where are the region that we can detect more virus RNA by using the spatial transatomics, and they are again, correlate to plasma cells and Tregs, right? So we go back to the protein levels. We first scan the viral antigen by using to detect the virus MP, because this is like more specific to the setting. Yeah. So as you can see, this particular patient, this is like their cancer tissue, and then we have a little bit of normal colonic crypts in here with normal colon structures there. You can see in here, and they are green color, meaning that they are positive for viral antigen. And you can see another crypt here. I hope you guys are seeing my mouse. So there's another normal colonic crypt here, which is actually negative. So it serves as a very good internal control. So we know that at the protein level, we can see this as well. And of course, we can also see some, the CD20 positive cells and the FOXP3 kind of like Tregs that surround within this area. And then we ask, this is it like it is just happen to be like that. So we look through the entire tissue and really cannot find any other part of the tissue that showing you the antigen positivity again. And especially this is a small piece of normal colonic crypts there, and the rest of the tissues are tumor, and we really cannot see any tumor region that have this. And this is kind of like validate our data in a spatial transatomic setting. And we still take it further. We actually look at RNA-scope to actually in situ popping this virus RNA, and we again in the adjacent normal colon, we actually can see these positivity cells, okay, but not the rest of the tissue. The rest of the tissue very, very rare. We can see that sporadic 1 or 2 cells there. So this is really giving us a confidence that this is real, and what we see in the spatial transcriptomics really can be translated into protein and also in situ setting. And not only that, we actually look at a few patients across multiple organs, and we can see this as well. And particularly, I want to bring your attention to the one of this interesting patient that we can see at the left bottom corner. This is exactly what I showed you just now. You have a tumor region that is negative for the viral antigen, but there is the next to it, the colonal crypts are positive. This is the same setting that I show you, and we can see in the spike antigen protein as well, protein levels as well. And the one that we send to RNA-scope that have positivity, we also send to RT-PCR to prove the positivity. So it's like modality to prove one particular evidence. So and later on, we also use another spatial transatomics platform to validate this. So we run through the immune, so-called COVID-19 public domain immune response signature, and then we found that this particular patient, there is one corner that very enriched. Again, it's correlated in concordance to the corner that we see just now. And then we run through the rest of the potential, like immune cells signature and some of the COVID receptors as well. And only the B cells and plasma cells you can see in here that enrich in the same or similar kind of region. That is really giving us another confidence levels. And look at the other patient, and then you can see that they are the enriched regions are actually across the very red particular regions, and you can see there are about 3, but the top right corners are we call it like still the focus area, right? And then again, the plasma cells and B cells again is very enriched in this particular focus area. And then if you do like unbiased or maybe arbitrary distribution into all the regions in this particular tissue into high COVID response versus low COVID response, and you can see this IgKC and IgHM genes representing the plasma cells. This is actually the immunoglobulin genes. Is really like showing a very significant difference in here, as you can see here in the diagram. So this is all the tissue levels information. So we are thinking, whether we can actually prove, like this, we can detect, yes, we can detect the immune enrichment here, and we can detect the viral antigen and RNA here. And is this immune response real and still there, So we actually design experiment to actually look at ex vivo, stimulation of this particular immune cells. These immune cells are extracted from, dissociated from the tissue that we show you just now. And then we stimulate them ex vivo by COVID peptide and see whether we can trigger something. First of all, usually this kind of stimulation is a mix. So you have myeloid cell, APC there, T cell, B cells there. So we want to first check whether T cells is really responding this. We see it. We see a lot of activation marker, granzyme B, CD38, and then we don't see it in a healthy kind of tissue. So this experiment is working. And later on, we actually run through a single cells experiment for this particular setting. So we run through single cell, we know every single cells of this one, and then we know, like there are, some of these are T cell, naive cells, regulatory T cell, B cells, and myeloid population. And we try to map this single cell back into our tissue, and we again see the B cells and Tregs again enriched in the region that we show you just now. And go back to the protein levels. And we show you just now the colonic cryptid positive for the viral antigen. And you can see that if you can appreciate some of these particular cells that next to the crypts, which is the epithelium, you can see them, they are also green color. I mean, of course, they are colocalized with the red, so they looks a bit yellow. So what are these cells? This looks like immune cells, actually. So we stand, of course, we show you the final outcome here, but we, of course, screen through a few of the candidate. And at the end, we know that these are CD14 positive, but not CD3 positive. So they are like myeloid and monocyte kind of lineage. And then we also know that they are CD68 positive, which is like kind of really macrophages, and they are SCE2, which is the viral receptor positive as well. So this is like, not surprised, because I think some reports also show that the virus can directly infect the macrophages. This could be one of the, the mechanism that why the virus can go through the whole body because of the APC or this thing. And we, again, sometimes we are trying to challenge our own data. We use multimodality. So we not only see it in the protein levels, we also see it in the RNA score in situ level. So this is gene level. So the gene, we also detect the virus and RNA together with CD68 and SCE2 on the same cells that you can see here. So, and at the end, we run through, like another cohort, which is the Postmortem cohort. We actually have about 20-somethings patients, and then we know that there are multi-organs also can be detected this particular viral positive macrophages or dendritic cells, and they are everywhere. So as a brief summary, spatial transatomics is really a good technology to actually, complement the current spatial technologies that we actually very, very exciting, and it's a game changer for studying the immune microenvironment. So for this particular study we actually show that we really can detect residuals, SARS-CoV-2 viral antigen, RNA, even the patient have survived and recovered from COVID, even up to 3 months. Okay, so and then we believe the immune response that we detect here, because we can prove it in the ex vivo setting, they are real, and multiple papers in this line actually have been impressed or accepted in principle. So particularly, I want to take another 5 minutes or so to actually show you the second story about the HIV co-infection. This is actually a paper that just got into gut recently. So, we always, wondering, the immune suppressed kind of setting, whether this will make the COVID make the patient more, susceptible for COVID, or even they get COVID, whether this will be more likely to be severe cases. So our collaborator from China, they actually, when this pandemic first started in Wuhan, they actually found some of this patient, the first batch of patients that HIV positive. So and then they see that some of these patients actually survive very long compared to the same batch of the other patients. So they are wondering why and how. So they are collaborate with us to actually look at whether we can see this in the immune setting. So some of this tissue we send to bulk RNA sequencing. This particular study is really like a dogma kind of a very good setting that we really have bulk RNA, spatial, and protein, the kind of thing I will show later on. So the bulk RNA sequencing, we actually can see that the COVID and HIV core infection, and they are very different from the COVID-19 alone kind of setting. And this only happened in the liver and kidney, but not so much in the lung setting. The lung is like, we don't really see a huge difference. And interestingly, if you do a GO of it, you actually can see that in the liver and kidney, you see a lot of this the T cells, reduction, and the macrophages are kind of like activated. But not this, again, the phenomena cannot be reproduced in the lung setting. And then, of course, we will do the spatial transatomics by using, nano-string DSP. And in here, we actually, select, this region of interest based on, again, CD3, CD68, and also cytokeratin to actually show the morphology. And we later on, I'll show you, we will analyze the CD3 rich and macrophages rich region separately. And in here, the key thing in here is that if you can see across these three organs, only the T cell reduction and the increase of macrophages can be only seen in liver and kidney, but not in the lung. And the rest of the immune cells, such as B cells and NK cells, remain no change. I mean, we don't see a significant difference between these two populations, and we don't see others immune cells. Again, this is the data for spatial transatomics, and we deconvolute into by using cybersort and into multiple immune cells. And the others immune cells in here is just like the rest of the minor population. And we want to see this in the spatial proteins levels as well. So we stain our multiplex IHC, and again, you can see that, the CD3, if you look at CD3 and CD20, CD206, which is the representative macrophages, and in here, you can only see this reduction of T cells and increase of macrophages in the extrapulmonary tissue, such as liver and kidney, but not in lung. This lung is actually sometimes even showing the opposite kind of setting. And if you go back to the bulk RNA sequencing data, you can see that the liver and kidney showing a lot of these macrophages enriched genes, and all this like activated T cells genes that is actually downregulated. And if you can look at the general bigger pictures, it's also showing the same. Kidney, liver showing the myeloid cells activation, involvement in immune response, but not in the lung setting. And just to recap, this the DSP setting is you have morphology, and then you select region of interest, and then you get the data 20,000 genes, right? So in here, if you look at only the CD3 rich region, and then we look at liver, in here we found that the liver is really interesting organs in this particular cohort of patients that co-infected with HIV. And you can see that the T cells abundancy positively associated with the viral load. Okay, the viral load is actually correlated to the T cells only in the liver and only in the non-HIV setting, because we know that HIV is a little bit, it's like basically targeting the T cells, right? This is a CCR5 mechanism, right? So it's actually making sense, right? So for the non-HIV patient, we can see that T cell is still functioning, is still responding to this one, but not the HIV setting. And you can see that in this one as well for the genes levels classification from the DSP. And also, interestingly, for the HIV setting, you can see the macrophages rather than the T cells, they are the one that correlate to the viral load. So this basically gives us like a hypothesis that the macrophage is kind of like compensating the T cell impairment in the HIV patient to fight the COVID. And this may be correlate to why they are surviving better in this particular cohort. So again, we are not trying to draw too much attention and conclusion from this small cohort for biological and clinical. We're just trying to show this to the public that this is a first batch of the patient in Wuhan, and this is like some of the autopsy sample that they collected. It's a very precious kind of medical clinical information that we want to share to the public. So again, at the end, we also show this in the protein levels that we stand multiplex see. We see the viral load is actually the green color is really associated with the macrophages percentage, and these macrophages are also expressing the activation markers such as CD80 and CD86. So again, I would already show you, we think that, usually people think that, in a COVID or even in HIV alone setting, their macrophages, all these can be impaired. In a HIV setting, in fact, if you read through, there are many of them actually reporting the macrophages can be long-lived, resistant to actually the cytopathic effect. But, in here, we are just trying to hypothesize that they are probably compensating the T cell to fight the COVID. So this is my last slide, just to actually show you one of the website and portal that we make through, and to actually show to deposit the public data, and to feel free to scan through the QR code. I'd like to acknowledge our pathology department, National Cancer Center, and some of the collaborators that and overseas collaborator from China, especially to actually share with us the precious data and tissue for studying the autopsy samples. Thank you very much, and I hope to welcome any question. Thank you.
Thank you so much, Dr. Yeong. That was a really fascinating presentation. We had a slew of questions that came in through the chat, so hopefully we can get to them before you have to run off. So one of the questions that came through from the beginning of the presentation, when you looked at the biopsies from the cancer patients, were any of those cancer patients on any immunotherapy? And do you think that would have some, correlation with the fact that they did, end up surviving COVID or not?
Sorry, Isabel, can you repeat the question? I mean, like a little bit soft here that I can't really catch the first part of your question.
Oh, yeah, sure. I will repeat it. So in the initial biopsy samples that you got from patients that have cancer and happen to have COVID, were any of those patients on any targeted immunotherapies?
No, they're probably having certain kind of treatment later on, but this particular tissue that we got for the surgical samples, they actually are considered treatment naive because they just get the surgery and then get the samples, and then we study the samples. But usually these patients later on, they may get some treatment, but it's actually after the surgery. It's a post-surgery setting, yeah.
Okay. And then a follow-up question to that came in from that part of your presentation. So in those patient biopsy samples, when you did the GeoMx Digital Spatial Profiling, you mentioned that there was the presence of some Tregs in the tissue. Do you think that is, just indicative of an immunosuppressive environment that is caused by the presence of the tumor, or do you think that having the Tregs there actually helped these patients recover from COVID, now that we know, looking back at some, severe cases of COVID and cases where people have actually passed away, that sort of a runaway overactive immune response causes a lot of the tissue damage, ultimately death.
Yeah, in fact, I think that is exactly what you say. I think we believe the Tregs probably is like kind of like a police officers that actually come in to actually make the particular region that infected by the virus, and they try to calm down the immune response and cascade in there. This is just our hypothesis. We do not really have a proof at the moment, but Tregs is also something that very generally. I would think that difficult to study in setting because as you can see, we show you more on the B cells and plasma cells setting is like the signature is more stronger, but the Tregs, because it's part of the T cell, sometimes the signal can be a bit noisy. So so we again we have the luxury to show you in the multiple modality kind of setting to actually prove one point. But that is again, you know, the thing that we want to promote, right? So if you want to adopt something like this, it's good to actually show it in different angle and pointing to the same direction. That is make the evidence stronger and convincing.
Thank you. So another question came in during the middle of your presentation. Could you comment on the workflow for the GeoMx Digital Spatial Profiling and how it might compare to other spatial transcriptomic methods that you have been exposed to?
I think we actually I actually didn't show that slide. I have a slide actually showing one of our paper. In fact, we actually have a review paper last year to talk about different platform on spatial transatomics, the pros and cons, the kind of thing. I think the key thing I would say advantages of NanoString DSP is actually the technical barrier is lower, I feel. It's easier to actually perform the experiment, and if you get the machine, is actually you probably easier to actually do that compared to the rest of the technologies that require a higher technical barrier. And the other thing is also the DSP can also do protein level. So I guess at the moment there is no other platform that can do proteins and the RNA side-by-side kind of setting.
So another question that came in towards the end of your presentation on the HIV work, which by the way is really fascinating. So do you think that there can be anything that can be extrapolated from that data, if at all? And I know it's a small sample size, to suggest that a robust innate immune response is very important, in preventing severity of COVID-19, disease, as well as, an early, death of some patients. I particularly want to add to this question too, that I found it really interesting, that there was, and obviously, with an HIV patient, they're going to have a lower T-cell response because of the nature of the HIV infection. But,
yes.
Do you think there's anything that can be extrapolated from that about, the importance of the innate immune cells being activated and being functional, and, kind of sounding the alarm first and taking care of a lot of the SARS-CoV-2 viral infected cells before even T cells and B cells get involved?
Yeah, I think first of all, I mean, as you say, I mean, this is a small cohort, and it's actually only from Wuhan at the very beginning of the pandemic, and whether this is really means something to the general population, at the moment we are not very sure. But if you look at it, I would think that this data sort of reasonable as well, because we all know that the one of the complication and one of the severe COVID setting, the mechanism is because of the cytokine storm, the immune kind of like overreaction, the kind of thing. So it's not really bizarre to actually see that some of this immune suppressive setting because of this one, they probably have a better outcome, or maybe not so severe, the kind of thing. I don't think this is something very difficult to actually conceive. What we actually want to bring out in here is that, so you can see this particular immune response or immunity that we discover very strong in liver for this patient, but not so strong in kidney, but still quite okay. Compared to lung is totally not in this kind of theory. So we not very sure in the multi-organ setting, different immunity from different organs, how do you actually really directly contribute to the systemic or general health of this particular patient. We are just trying to report that there is this thing, and require further study to understand how actually this is going to contribute for the infections, and also later on maybe vaccines kind of setting.
Excellent, and that's a perfect segue to the last question, which is related to vaccines. So now that we're seeing a massive rollout of, vaccinations for SARS-CoV-2, and reports are coming out of certain side effects, and potential rare cases, where there could be, severe side effects. How do you think spatial transcriptomics can be used to resolve some of these complicated cases of, potential side effects with a vaccine? And along with that question, also, what is the next direction of your research, that you're going to focus on with the spatial transcriptomic tool?
I think there's thanks for the question. I think that is actually something that we are definitely interested to study. I mean, I think there are some groups already started to collect some of these samples. It could be the same setting, right? If you go back to the slide that we show you, we have postmortem setting, and which is the decedents, and also the survivals, right? So in the vaccinated setting, it's also the same. So there are people that after vaccination, they actually went for surgery, so we can get this kind of tissue. And some of these patients, may not directly because of the vaccines. Some of these patients actually, or some of this healthy donor, they actually having certain kind of accident or something. So they have the postmortem samples. So I think the digital spatial profiling technology can be applied to study this kind of cohort and to understand. I guess the key question at the moment first is that to understand the viral distribution. Of course we can learn this from the blood, but the organ specific setting is actually I would think this is a very important piece of information for the scientific community. So this is number one. And number two is if you detect this virals, either RNA or antigen, and their correspondence, immune response, and what are the immune cells, and what are the immune genes that enrich in those regions, and whether there is any organ specific kind of setting like I show you just now, it will be a very important and precious scientific information to actually help us moving forward, maybe preparing for the next pandemic, if there is any. Hopefully there's not.
Well, thank you so much. We really appreciated the talk. Oh, it looks like there's one more question that is coming in really quickly, and hopefully we can get to it before we have to wrap up. So do you have any plans, or do you have access to any samples of, patient biopsies of, say, solid organ transplant recipients who have contracted SARS-CoV-2 to understand what is the effect, if any, of people that are on long-term immunosuppressives and how that affects the outcome of COVID-19?
I think at the moment the short answer is no. But I guess if you really want, we can approach some of the collaborators for that. And of course, in the audience, if anyone want to, work with us, collaborate with us on this kind of samples, we are more than welcome to help.
Excellent. So on that note, I'd like to thank you, Dr. Yeong, for joining us for the excellent presentation. And also I'd like to again thank Dr. Eugenia Ong, who was on earlier. And again, for those of you listening to this LabRoots Coronavirus virtual seminar series webinar, if you have questions that didn't get answered, the folks at LabRoots will follow up. And again, thank you so much for.
Thank you.